All right, hasn't stopped on me yet. The big picture. We're going to continue with this big picture. We already know the 4 tissue types: connective, epithelium, nervous, and muscle. And now we're going to, for the rest of this block, just go through all of the systems that we have and really talk about the histology of all of the organs. And then by the end, you should be prepared, hopefully, to take that shelf exam. Okay. So here are your learning objectives. Those are your learning expectations. So the respiratory system encompasses several organs, not just the lung. We're also going to talk about the pathways that air got to the lung, starting with the nasal cavity, and then the pharynx, and then the larynx and the trachea, and then we're finally down to the bronchi and into the parenchyma of the lung. Now, there are two, I did want to point out that we're not going to talk about the mouth. Now, you can certainly take air in through your mouth, but we're going to cover that with the digestive system. Okay, so the respiratory system has 2 parts to it, if you just sort of divide it anatomically. And one part is called the conducting portion, and that's going to be the part of the system that brings air to the not only to the lung, but even to the smaller and smaller air passages, where you're not going to have any gas exchange occurring in these structures. And then there's an anatomic shift over into the location where you have alveoli, where gas exchange is going to occur. And so we call that part the respiratory portion. But most of the respiratory system, different anatomic structures we're going to talk about are going to be differentiating the conducting portion, but if you looked at surface area, the alveolar surface area is much larger. Okay, so most of the respiratory system is going to have an epithelial layer that has 5 different cell types in it, and we call it respiratory epithelium. Okay, the most common cell or most prominent cell in this epithelial layer is a ciliated columnar cell. And so that cell has a lot of cilia, we'll talk about in a second, and just collectively it is pseudostratified. It looks pseudostratified in most places. So that means pseudostratified, that it's really one layer, but they all look like it's more than one, but all of the cells touch the basement membrane, and that's why they call it pseudostratified. And so in this picture here, you can see the respiratory epithelium. You can hear a dog barking maybe, but here's the respiratory epithelium, and there are lots of nuclei in this epithelial sheet, and there's cilia here at the apical portion of the cells. Now, underneath the epithelium sheet is where the basement membrane is, and it's pretty prominent in the respiratory system. And underneath the basement membrane, there is an area of connective tissue that we refer to as the lamina propria. And this region has some special structures throughout the respiratory system in it. And for example, glands and cartilage would be 2 things that would be in that area. And so that term is kind of thrown around a lot in this chapter, and they don't really explain it too well, so I just wanted to point that out. Okay, so the ciliated columnar cell is the most abundant. It's got lots and lots of cilia on each surface, and the cilia is going to rhythmically beat. So they're motile cilia, and they are going to bring mucus and other particles that get trapped in the mucus up out of the respiratory tract so that it can be swallowed by the pharynx. Here is another picture of that displayed here, so you can see the pseudostratified columnar epithelium, really nice line of cilia here. Okay, and you can also see the basement membrane. This is a trichrome stain, so it's stained blue. All right, the next cell is pretty abundant actually in a lot of parts of the respiratory system, is the goblet cell. And we talked about goblet cells early in the class. They are unicellular glands, so they are going to produce mucin. Mucin will mix with water and become mucus. And you usually see the mucin inside the cell towards the lumen of the epithelial sheet, and it's filling up the cell, and the nucleus gets pushed to downward and kind of shoved out of the way. Actually, this is the nucleus, sorry. Pushed downward and shoved out of the way. So in the arrows you can see all the goblet cells. There are no cilia at the end of a goblet cell, okay? But they can be very abundant in some areas, like I'm about to show you. And sometimes you could see it just looks like it's pale staining, so it's not staining really deep. You're not necessarily going to see granules inside, so that's why it has such a light appearance. So in this larger image, I wanted to give you something clearer and a little bit more zoomed in, and you can see multiple goblet cells here all throughout this epithelial sheet. So they're pretty prominent. All right, brush cells is another cell type. They're not mentioned in the Weeder's book for lab, but they are talked about in Juncaris. They are columnar, but they do not have cilia on them. They have microvilli, and there's only about 3% of the cells in the epithelium are brush cells. So there's not a very big cellular population. But they don't have cilia, so they're not doing anything with moving. But they do have chemosensory receptors on the end of these cells, so it's kind of like the gustatory sense of taste. And these cells have adjacent nerve endings, afferent, so sensory nerve endings that are adjacent to the basal surface of the cell. And you don't have to identify these, but you can see in this picture here, this is an EM from your textbook. I'm going to zoom in on it in the next slide so that you can see some of the features. You can see the ciliated cells, and you can see cells that have mucin at the apical part that's sort of bulging out here. And then you can see these cells over here have microvilli on them, blunt microvilli instead of cilia, and those are going to be the brush cells. And then there's another population called small granule cells. You don't have to identify these either. They do represent about another 3% of the total cell population. And they are part of the diffuse neuroendocrine system that exists in the respiratory system, also exists in the digestive system, okay? And so you're not really going to see them or have to look for them. I had to actually search pretty far to find an EM of them. Of these types of cells. And just in case you're really super interested in small granule cells and want to learn more, I put a link into this article here just in case you want to kind of get a gist for what they are all about. Okay, now then there are basal cells. Basal cells are small spherical shaped cells that lie adjacent to the basement membrane, and they are going to be the stem cells for all of the cell types in the epithelial sheet. And this is a very common pattern that we're going to see throughout several systems that have stratified or pseudostratified epithelium, or epithelium that changes like a transitional. And so these basal cells are sort of kind of, I guess, more like a universal type of cell that'll be in lots of epithelial sheets instead of just targeted to the respiratory system. In this picture here, I pointed out several basal cells so that you can see where they're located at. The nuclei are going to be the most basally located. Okay. Now, not all of the epithelium in the respiratory system is olfactory. So I meant to say respiratory. We do have a couple places where the epithelium is responsible for transmitting the special sense of smell, and that is going to be over the superior nasal concha and the roof of the nasal cavity on both sides. And so and the arrow is kind of just pointing to that. And it kind of makes sense because here's the ethmoid bone, and on the other side is where the crista galli is and the cribriform plates are, and that's where the olfactory bulb sits in there. And so with this epithelium, you also will need to identify. It kind of looks similar, but when you really get the hang of it, you'll be able to differentiate why it looks different. It has completely different cells in it. It has supporting cells, which are columnar cells with microvilli, and then it has olfactory cells, which are actually bipolar neurons, and it has about the same population for both. And then there are basal cells that are adjacent to the basement membrane that are going to give rise to both the supporting cells and the olfactory cells, or the bipolar neurons. You're not going to see any goblet cells in the olfactory epithelium. There's not going to be any mucus that is deposited over this specialized epithelium to prevent binding of the odorous substances to the cilia. But there are specialized glands in the lamina propria called Bowman's glands, and you can see the lumen of the glands all positioned here, and I'll show you a duct in a little bit. And then you also have very large bundles of axons from the afferent nerve endings for the sense of smell that are going to be in the submucosal area. Okay, so the nuclei are positioned in different locations of the supporting cells and the olfactory cells. The kind of line that you see here that is towards the lumen of the nasal cavity, these nuclei all belong to supporting cells. This is where the nuclei kind of lie in this decently organized line. Okay? And they don't really know a lot about their function, but they know that they have a lot of ion channels and that they really do help maintain the microenvironment that's conducive to the sense of smell. And then the olfactory cells nuclei are located here at this level of the epithelial sheet. Okay. And then the basal cell nuclei, when you see these closer up in lab, you'll see there's a row of basal cells that's adjacent to the basement membrane. And then you can see other components, the Bowman's glands and the nerve bundles, or axons. Okay, so this is a scanning EM of the apical end of an olfactory neuron, and you can see the cilia projecting outwards. And I really liked it. I don't know, it just sort of gave me a better kind of feel for these olfactory neurons to really sort of get it. I just liked it. I'm just gonna leave it with that. And so the Bowman's glands are going to be putting this fluid out that's going to continuously be rinsing off and bathing the cilia that are projecting from these olfactory cells. And so these cilia are going to be ready for stimulation. And so when you have odoriferous substances that are in the nasal cavity, they can bind to a lot of these transmembrane chemoreceptors that are sticking off the ends of the cilia. And so that's what I'm trying to show you, this pretend, odoriferous substances, molecules bouncing around, and then some of them are going to come down here and then are going to bind to these transmembrane chemoreceptors. And that is going to simulate an action potential that is going to extend downwards. This is like pretend this is the end of one of the cilia, and it's going to extend downward, and it's going to actually cause an action potential that is going to be transmitted through an axon, and it's going to synapse with another neuron in the olfactory bulb, and you'll learn about the rest of the track later. But so this is where the cristagalli is, and you can see all the axons of the olfactory neurons projecting through the cribriform plate. I meant to say the cribriform plate. And it's really neat when you think about it that these neurons are replaced about every 2 to 3 months. And so here we have a place in the body where neurons regenerate, which is a very rare thing for our bodies. Okay, and then I liked this picture that I saw online where it showed a duct from a Bowman's gland coming through the epithelial sheet, and so the fluid that is made in these glands can go right on top of the epithelial sheet. And so that constant flow is really important for maintaining the patency or activity of the chemoreceptors. Okay, so the nasal cavity is going to look unique depending on the orientation that it is in. Like this is a horizontal cut of the head, and actually I think this is a cat. And so you can see that it doesn't look, it almost looks upside down of what you would think that it should look like. And when you zoom out and you can see these bones here, you'll kind of get an indication of the orientation maybe a little bit better. I think what you probably are more comfortable with is a nasal cavity cut that's in a frontal or coronal section. So that's displayed here. And this is will also be in your lab set. So I just wanted to point out that the nasal conchae and the way that these, the walls and the orientation of the nasal cavity could just be very diverse. Okay, so don't forget the nasal concha are those projections from the lateral wall of the nasal cavity, and they're made up of bones. The superior and middle nasal concha are part of the ethmoid bone. I'll show you here. This is the human diagram of a human. And then the inferior nasal concha, remember, is its own bone within itself. Okay, and these bones are going to be lined with mucosa. You probably remember taking off the mucosa when you dissected the nasal cavity. So that's what we'll be looking at in when we look in lab. Okay. The nasal cavity mostly has respiratory epithelium. A little bit inside the vestibule of the nasal cavity is where the kind of skin from the ectoderm kind of wraps in. And so you do have a little bit of thin skin at the ridge of the vestibule of the nasal cavity, but most of the rest of it has respiratory epithelium. Of course, you're going to have a little bit of that olfactory epithelium I almost said northernmost part, but the most superior part of the nasal cavity. And then a cool feature, and your book really doesn't talk about it this year, this new edition doesn't go over it, but I still think it's cool. They just say that there's complex vasculature just deep to the epithelium. So here you can see the epithelium and these very, very large thin-walled vessels that fill in the submucosal area. And it's important for warming and the air that is inspired through the nasal cavity. I have also other textbooks that call them swell bodies. I don't know how common that term is, but that's all of these vessels here. And if you look at them kind of closer up, you can see that they're thin-walled, and your textbook actually describes it more like arteries, but I have also seen them described as veins. So I changed the term and just put vessels because I don't know if either of them are correct. Like when you talk about cavernous spaces in erectile tissue, there's not an artery, or there are arteries, but the tissue itself that is cavernous is just vessel-like structures. And then in this slice, you can also see bone. That's part of the nasal concha, and you're going to be looking at bone. It's going to help orient you, and so we are going to have to get a little used to its appearance. Okay, and this is a slide of respiratory epithelium. The nasal cavity is going to have goblet cells. It's going to have ciliated columnar cells, and you will see glands and the submucosa. So those are features that you're going to look for identifying the respiratory epithelium. But if you look over to the area where the olfactory epithelium is located at, then you will see a few differences. So the epithelium, see all these lots and lots and lots of dark nuclei, and you don't see any goblet cells here. I know this kind of looks messy, but and so it's kind of hard to see whether or not what kind of cilia they are. So they both kind of look ciliated, and the olfactory epithelium has cilia, they're just not motile cilia. And then you can see Bowman's glands here, and those are going to help point out that it's not respiratory, and then these axon bundles. And here is another slide of olfactory epithelium from Tulane, and I just wanted to show you the tissue looks much different due to the different staining, and we'll be checking this out in lab. All right, now we're going to go through the rest of the structures of the respiratory system and talk about the microanatomy of each, which are going to be changes in the epithelium and changes in the amounts of smooth muscle and cartilage and glands in the lamina propria. All right, so the conducting portion is going to include all of those structures, and the respiratory portion is going to include when we start talking about respiratory bronchioles and alveolar ducts and alveoli. Okay, so when you're looking at a slide, you're going to have to look around and pick up the features. The epithelium, because respiratory epithelium is prominent, it's not going to be the only thing that's going to help you, and sometimes it's not even going to clue you into anything except for the fact that you're in the respiratory system, period. So don't forget that you are also having to identify other structures that are there. I'm not going to let you off the hook for identifying blood vessels and nerves and glands. Cartilage, bone, and muscles. So those are all going to be players in this system, and a lot of the slides will have all of those things on it. Okay, so the nasopharynx is going to have respiratory epithelium and looks very much like other places in the where the nasal cavity is and lower down. But it does have a special region where you have the pharyngeal tonsil, where you're going to see big aggregations of lymphocytes underneath the respiratory epithelium. And so that's what you're looking at here. And Dr. Glover will talk to you about the cells. I think she's actually talking to you about them tomorrow. And I'm going to get you down that path because your immunology block is starting. All right, the larynx, hopefully you haven't completely forgotten that the larynx is made up of a bunch of cartridges and also has overlying muscle. And so hopefully you haven't forgotten that. Also, just anterior to the muscles and cartilages of the larynx, that's where the thyroid gland is going to sit. And it has a lot of glands that you couldn't see before in the lamina propria, but you're going to see now. If it's cut mid-sagittally, it definitely looks different because this is cut in half, and so you're looking at this is superior, this is inferior. And this is towards the inside of the larynx, and this is on the outside. And so you can see the fold. This is the vestibular fold, and here is the vocal fold here from a distance. And you'll be able to zoom in and identify not only the epithelial transitioning that occurs, but muscles, cartilage, and a little bit of the cartilage is actually ossified in this individual. Okay. It looks very different in a cross section. So here you can see the cartilages, and you can see lots and lots of glands here. I can see from this magnification that we have some glands and loose connective tissue in places, and we have muscles in other places. So that's what you can see, and we'll be able to zoom in. I have them labeled here: cartilages, epithelium. There's a thyroid gland. I said glands. And then we can have different muscles, and we will see other glands as well. Really cool thing is that the larynx, you're going to see not only respiratory epithelium, but you're going to see a transition to the stratified squamous epithelium over the true vocal fold and also over the epiglottis. And so we get a little more friction in those areas, and we want to protect those structures from the constant abrasion of fast-moving air. And so that's why we have the transition to stratified squamous. And this is just a little bit of exactly what I just said. Okay, and this is a close-up picture of a larynx where over the true vocal fold where you have stratified squamous non-keratinized. And this is the same larynx, but respiratory epithelium where you can see prominent goblet cells. Even if you can't really see the cells close up, the goblet cells are telling you one of two things. You're either in the respiratory tract or you're in the digestive tract. Okay, and you'll learn later that these cells are not columnar enough. A simple columnar is the digestive tract, and so this is a stratified or pseudostratified. All right, the trachea. The trachea has some prominent features that really stick out. It's got this nice big C cartilage ring that you can see that kind of clues you in that you're in the trachea. So it's an actual tube that's cut and on the slide instead of that funky looking larynx, and so that kind of helps. All right, if we get in closer, we can see that this structure has respiratory epithelium here. And then it has a lot of glands in the lamina propria. So they're really much more pronounced than any other place in the respiratory system. And then if you have a low power view, being able to see the C-shaped cartilage really helps give you an indication that you're in the trachea even before you confirm it by looking at the other features. There is an adventitia around the whole organ, and then the trachealis muscle, which is really not depicted well because it's not cut right through the trachealis muscle in this slide, but I have it on the next one. But there is also an extension of the perichondrium that is going to be fibroelastic and is going to go from one side to the other of the cartilage that gives the trachea rigidity and strength. Here is another picture where you can see the trachea, and you can see the trachealis muscle going from one side to the other. And it's just next to the esophagus, adnexal. All right, so this is the trachea in the Michigan slide, and so you get to see the bright blue basement membrane because it's full of collagen. And you can see prominent goblet cells in this stain, so this is something that you'll see in lab. The next structure is the bronchi. Bronchi can be primary, so it could be outside of the lung, but most of the bronchi that you're going to see histologically are going to be intrapulmonary bronchi, so within lung tissue. And so you'll see lung tissue, and then you have to sort out where the bronchi are positioned relative to the other structures. The bronchi are going to have respiratory epithelium And it's going to have cartilage, but the cartilage are not going to be big and C-shaped like the cartilage was in the trachea, but rather it's going to be smaller plates of cartilage. And as smaller the bronchi get, smaller the plates of cartilage get. And there are still some seromucous glands in the submucosal area. And so you will be able to see an occasional bundle of glands, and you'll be able to see some cartilage. And then we start to see smooth muscle that shows up in the lamina propria as well. So the lamina propria is thinner than in the trachea. It's not going to have as many glands, but it is going to still have cartilage in it. All right. And then you see the lung around it and sometimes some lymphatic tissue. So in this slide, here's a bronchi here, bronchus here. And it's not primary because it's within the lung, so it has to be at least secondary. And then here you can see a smaller bronchus, maybe that's a tertiary, not sure. And then you can see a vessel here, and you can see what's probably a vessel here. I don't see any cartilage in the walls of this, and I can't see it close enough to know, but I think it's a vessel. This has got red blood cells inside. Usually the arteries run along with the air spaces, and the veins run along kind of a little farther off inside the lung parenchyma. But this could be cut obliquely. All right, if you get a closer view of the bronchus, or the wall of the bronchus, I can see the epithelium here. So this is nowhere near as thick as the respiratory epithelium was in the other parts of the more upper respiratory system. This is definitely starting to get shorter in its height. And then for the first time, you get to see smooth muscle here. It's a pretty decent band of smooth muscle that shows up, and constriction of the smooth muscle is going to cause narrowing of the airway. And then here you can see this really nicely stained cartilage. You're going to need to identify cartilage in this block here. You're not going to necessarily know all of the little types of cartilage. You just have to recognize it as cartilage, because Dr. Gregory is going to talk about the skeletal tissues when she does bone. Okay, and then here you can start to see some alveolar spaces, and you can see they are abutting the wall of the bronchus. Here you can see the epithelium closer up. They still have cilia on them. They're nowhere near the stratified nature as the previous epithelium that we had seen in the respiratory system. So don't forget the lung is segmented into primary, secondary, and tertiary, and this is just degrees of branching. So you have one on each lung of primary, and then they're going to branch, and they're going to serve, when they're secondary bronchi, they're going to serve as one to each lobe, and then when they branch another time, they are going to serve all individual bronchopulmonary segments, and those are the tertiary bronchi. So they all have the same characteristics that we just discussed. It's just that they're going to be progressively smaller. And so this is going to be a bronchus. I only need to know that it has cartilage here in the wall. These look like they could be little pieces of parts of glands, and so that those two things together, I can also see smooth muscle at this magnification, but I don't even have to look at the epithelium because I can see that I'm within the lung. I see cartilage, so right now I know I have to be in a looking at a bronchus. All right, and that's because the next level, which is a bronchiole, is deficient in any cartilage. It has no cartilage in the wall, and it has no glands in the submucosa. Okay, so that's what makes it a bronchiole: the absence of cartilage, the absence of glands, and this is a progressively smaller airway. And so here you can see a bronchiole within lung parenchyma. You can see two blood vessels depicted, 1 and 2. And here you can see a whole bunch of, there's some vessels kind of scattered in here, but generally speaking, there are a lot of alveoli. Now, the epithelium gets even shorter here, and it can even end up just being simple columnar in its appearance, okay? Here is another picture that shows you a longitudinal, longitudinal cut through a bronchiole. And so you can see the bronchiole ends. It ends in the bronchiole that they refer to as a terminal bronchiole, means the ending of the bronchiole, and it's going to open up into the first part of the respiratory portion of the respiratory system, which is a respiratory bronchiole. And I know that that is the case even from this magnification, because I can see that this part of the, I can see that this is a bronchiole and it's continuous, and then this part gets broken up and I can see it open into a couple alveolar spaces. Okay, and you're not gonna have goblet cells at this part of the respiratory system, so you are going to lose that clearing function that mucus was serving. However, you don't have you're also going to start losing those ciliated cells, so you're not going to have them to get mucus out of that area if it did have mucus. Because the epithelium is going to change from a ciliated columnar, mostly in nature, to a new cell called a club cell. A club cell, which was previously called Clara cells, they are dome-shaped, they don't have any cilia on them, and they make lots of secretory granules, so they're usually kind of paler in appearance. The cool thing is that they can divide and they can replace other cell types that are in the alveolar spaces called type 1 and type 2 pneumocytes. So they secrete surfactant components, and they also have enzymes, so they can detoxify noxious substances that have been inhaled and have gotten down into that part of the respiratory system. So you don't have goblet cells, you don't have cilia to remove particles by the escalator that gets the mucus out of the respiratory system, but these cells can actually help detoxify some substances, and they will also stimulate your immune response by releasing cytokines. Okay. All right, now the lamina propria that of a bronchiole has a lot more smooth muscle in ratio to the thickness of the wall. And so this smooth muscle is going to be autonomically innervated and under control of sympathetics and also the vagus nerve. And it's kind of counterintuitive maybe, but the sympathetic nervous system, stimulation of the sympathetic nervous system causes relaxation of the smooth muscle cells in the walls of the bronchioles. And that's because if you had a stress response and you needed to fight or fly, you would need more oxygen to your lungs to support you in your physical activity that it was going to occur. And so it might be kind of counterintuitive, but so it's one of those things kind of like pupil dilation and constriction is. And so this is another location where it might not be the way that you would expect it to be. And as you'll learn in your physiology class, that this constriction that can occur from the smooth muscle cells in the wall contracting will increase airway resistance and will become, really problematic for the person who's trying to breathe air in and out of their lungs. And so Dr. Levitsky will talk to you more about that. All right, and so the respiratory bronchioles are going to be where you have a continuous terminal bronchiole start to break up and open into alveolar spaces. So at this point, this terminal bronchiole has become a respiratory bronchiole. And you still will see a couple scattered epithelial cells that look like club cells, and that's how you know it's not just an alveolar duct, because you still have a couple patches of these epithelium. And it's not super easy to find because of the way that these sections are cut, and they catch alveolar spaces and bronchioles and bronchi and all different kinds of orientations. So please be a little bit patient with yourself when you're trying to find this, okay? All right, and so the respiratory, this is from farther back. You can see there's still some club cells here and here. So in this area here where you have opening up into alveoli, you have a discontinuous bronchial epithelium into the alveolar epithelial cell types on the walls. This area here is a respiratory bronchiole, and that's the first part of the respiratory portion of the respiratory system. Okay, now for the main event, alveoli. It seems like you talk about alveoli so much in like the physiology class, and then when you get to histology, you talk so much about the respiratory system overall, and then only a little bit of time about alveoli, but that's okay. I want you to understand this conceptually so that you can really put everything together. So the alveolar air spaces have three parts to it. It has an epithelium, the surface epithelium. All right, so we have new cells to talk about that we haven't talked about yet. Then there is what's called a fibroelastic and interstitium. So Levitsky will talk a lot about it just being an interstitium, or interstitial spaces or walls. And so this is the supporting connective tissue that is amongst or just at sort of like the basement membrane, but it's extremely, extremely thin. It's really, in a lot of places, diffused basal lamina. But it is going to still contribute to the structure and rigidity of the alveolar wall. So the walls of the alveoli is going to have some supporting connective tissue. And then all of these walls, I don't know if you can see from this magnification, but all of the little red circles, those are all red blood cells. So all of the walls are also made up of pulmonary capillaries. And they're going to be cut in all different orientations, which is going to be very, very maddening when you're looking at a histology slide. So this is one close up, and you can see the pulmonary capillaries sort of winding through. To me, I kind of feel like the pulmonary capillaries holding everything up, and I know they can't really be doing that, but I don't know, it just sort of always seems that way to me, because you only cut the epithelium in every once in a while in through a nucleus. But there is an epithelium, the cell membrane of the epithelial cells are lining all of the alveolar spaces, even if it doesn't look like that in a histology slide. So you're going to have the epithelial cells, you have the endothelial cell that is lining this pulmonary capillary, and then you have fibers, collagen, reticular, and elastic fibers that are also woven into this wall. That's collectively called the interstitium. Oh, okay. So it's kind of disappointing though to look at a real histology slide, but this is the reality of what we're dealing with. The lung is cut. It's like a honeycomb appearance, and it's a very, very 3D structure, and we're cutting it, and it's kind of shrunk down and collapsed, and so that's why it looks so abnormal. But you got to sort of use your imagination to kind of put it back into place so that you can figure out what you're looking at. Okay. So the alveolar epithelium is going to contain two cells, a type 1 pneumocyte, and most of them are going to be type 1 pneumocytes, and a type 2 pneumocyte. And so the type 1 pneumocytes are very stretched out, so you can't really even see them as cells, I think, a lot, unless you cut it through the nucleus. But the type 2 pneumocytes are not as stretched out, and so they're a lot rounder in shape, and that's they take up supposedly more of the cell populations, and they sort of take up the places where the alveoli come together, and they can show up sort of like in little bunches or groups. Okay, so the type 1 pneumocyte, like for example, here's a nucleus here, sort of like all stretched out. You don't always, you definitely have a type 1 pneumocyte along this entire border. Even if you don't have a cut through the nucleus, its cell membrane is still there. And that is the cell that the oxygen and carbon dioxide is going to pass through after it comes out of the endothelial cell that's lining the capillary. Okay. Then type 2 pneumocytes are harder to see on a histology slide, but we'll be looking around for some anyways. But when you look at an EM, they look really neat looking because they have these structures called lamellar bodies, and it almost looks like it's a layered, some sort of layer shell or rock. And they also have multivesicular bodies that are also here. They're a little smaller, and they look different in appearance, and there's one there. And they are actually going to join the lamellar bodies, and this is where you have the newly synthesized surfactant located at. So a type 2 pneumocyte is another cell that you have in the respiratory system that is going to secrete surfactant. And pulmonary surfactant is important for decreasing the surface tension in the alveolar spaces. And Dr. Levitsky will talk to you about that at length. And surfactant is going to contain an important phospholipid, and it's called DPPC. I cannot pronounce that word, but you give it a shot. And also several surfactant proteins. They're called SPA and like SPB, SPC, and SPD. And your textbook goes into detail about the different proteins and what they are specifically responsible for, if you want to read that information for its clinical relevance. But generally, you'll learn the physiology, and you now know that type 2 pneumocytes are the cells that secrete these. substance. And here you can see a couple. I just sort of pointed a couple out. I guess this is a pretty plastic section. Most of our lung sections don't look anything like this. But a type 2 pneumocyte is sometimes easier to find if it has some nucleoli in the nucleus. Okay, and so don't forget, it's not really something that's very easy to see, but the septa that are in the alveolar, between the alveolar spaces, are going to have not only collagen fibers but also a lot of elastic fibers and reticular fibers. The elastic fibers are very important for the recoil that occurs in the alveolar spaces and very important therefore for pulmonary functioning. And this is going to be apparent when you talk about conditions like emphysema. Now, there are also several different immune cells that can be found inside the interstitium of the lung. Notably, macrophages and mast cells are going to be there, but you can find even eosinophils and other cells there as well. This is a lung of a congestive heart failure patient, and you can see very, very congested pulmonary capillaries. Blood comes out into the alveolar spaces, and macrophages are going to ingest those red blood cells, try to clean up the debris, and so what we're looking at here are hemosiderin-laden macrophages. Macrophages in the respiratory system are commonly referred to as dust cells, and you can see them in normal circumstances, and not all of them look active. They just are sort of wandering around. So the big picture of gas exchange is that oxygen is going to come down through the conducting portion, down to the respiratory portion, and into an alveolar space. And I said oxygen, but I meant air that you inspire. So that air is going to have several different things in it, but one of the things that you want out of it is oxygen. So oxygen is going to have to pass through the endothelial cell, through the interstitium, through the endothelial cell. I think I said endothelial cell. I meant the epithelial cell that lines the alveolar space. So a type 2 pneumocyte, it's got to come through the interstitium. If it's got, hopefully it doesn't have a real thick interstitium. Sometimes with pathologic conditions like chronic bronchitis, that will get thicker. And then it has to cross the endothelial cell's plasma membrane and the cell itself and come in to attach itself to a red blood cell. And the same note, carbon dioxide comes out of the red blood cell and is going to come across the same barrier but the opposite direction and come into the alveolar space, and you get mixing of air as you inspire and expire to replenish the air to bring more oxygen in and more carbon dioxide out. So that's the big picture that you will be talking about for the next couple weeks. So that means anatomically, you end up with a very specific anatomic gas barrier. And this is not anything that is new to you. I just said that here's a pneumocyte type 1 cell membrane. Okay. This is the fused basal lamina that it has with the endothelial cell of the capillary. And so oxygen is going to come this way, and this is a erythrocyte, and it is going to attach itself to the red blood cell, and then carbon dioxide is going to go the opposite way and come into the alveolar space. All right. Do you remember when the lungs developed, and in gross anatomy we talked about the visceral and parietal pleura that were the coverings of the lungs, and then in between there you had a really small space that was called the pleural space, and it had a little bit of serous fluid that was secreted by the mesothelium that lined the pleura? Well, that's what we're going to see here. So the visceral pleura, which you get to see when you're looking at a lung, the parietal pleura it's been removed. You're not going to get a chance to see that in a histology slide. But for the visceral pleura, you can see the mesothelium because it's going to be a simple squamous epithelial sheet that we'll look at. And then right underneath that, you'll see a little bit of connective tissue. And that's kind of synonymous when someone talks about the serosa of a lung or the serous membrane of the lung. So these are all the anatomic structures that people are referring to. So in this section, you can see that this is the cut part of the lung that is continuous with the rest of the lung parenchyma, these areas here. This has got a little bit of a darker pink line that sort of comes all in this direction. So this is the site of the visceral pleura that we're looking at here. And if I zoom really close in on one, I found this, I don't remember if it was this slide or if it's a different slide, but I zoomed in so you can see the simple squamous mesothelium, and it's called mesothelium because of its derivation. That's on the outside of the pleura. outside of the lung, and then here you can see the underlying connective tissue. And so it kind of looks somewhat messy because of the artifact here, but it's continuous with the lung parenchyma. You can see alveolar spaces located